Okay, looks like everyone's in. Fantastic. So officially a good mid-afternoon, everyone, and welcome to the mid-afternoon masterclass as part of the University of Melbourne Science Festival and National Science Week. My name is Julia and I will be your host today. Before we begin, I do want to acknowledge country. I want to acknowledge the Wurundjeri people of the Kulin Nation as the traditional custodians of the lands on which I am on today. And also, of course, acknowledge the traditional custodians of the lands on which you, our audience, are zooming in from as well. I want to pay my respects to elders past, present and emerging, and extend that respect to all Indigenous people in the audience today. Also, really importantly, I guess at this time of National Science Week, I think it's really important to acknowledge and respect that Aboriginal and Torres Strait Islander people have been researching and learning and caring for this land for tens of thousands of years, and so they are our first scientists. So, mid-afternoon masterclass. If this is the first time you've been here, welcome. If you're back again, welcome back. This is a series of events that happens at 2 p.m. Every day during Science Festival, and we hear from a different academic every day talking about a variety of fascinating topics. So each session runs for about 45 minutes and it includes a short presentation as well as a Q&A as well. So you, the audience, can be part of the presentation as well. So today we are onto the weirdest mammal in Australia. So we're going to find out more about the monotremes and what we are starting to learn about their unusual reproduction. And to talk on this topic, I would like to welcome Dr. Jane Fenelon, a research fellow from the School of Biosciences. Hello, Jane. How are you going?
Hi, good, thanks.
Good. So I'll give the audience just a little bit of a background of your research. So Jane completed her doctorate at the University of Melbourne. She was investigating the ability of tammar wallabies to pause and restart their pregnancy, something called diapause, which sounds fascinating in itself. And frankly, that should probably be another presentation that we can do. Jane then continued this work at the University of Montreal over in Canada, but working with the mink and mouse, and then onto Ottawa Hospital Research Institute working on embryonic development. She was back here in 2018 and has been looking at the reproduction and development in short-beaked echidnas. So that's the overall view. Thanks, Jane. I'd love to throw to you firstly before we sort of get into the whole presentation and just find out what it is about this area, about biosciences, about monotremes, what it is about that fascinates you so much, and why did you decide to focus your research on it?
Well, I just always, from a very, very young age, always wanted to work with animals. And originally I was going to try for vet, but I didn't actually get in. And so I ended up doing a science degree. And then I discovered that I really like working with wild animals and just finding out about all the weird things that they have. Yeah, so I started off with, like, the first weird thing was the pausing pregnancy, which I'm still doing some work on. And now it's echidnas.
Fantastic. So I think, I mean, it's an amazing story. I think there's a number of different people that have tried to go into something and then suddenly they take a subject or go in a different path and realize that actually the other thing that they hadn't considered is what they actually love. So that's fantastic. So before we get started, I do want to let everyone know that there's a couple of ways that you can interact with Jane throughout this presentation. Obviously, we're on Zoom and on Zoom webinar, so we can't see or hear you. But there are two particular functions I want to highlight to you. We've got the chat function and the Q&A function, and you'll find those at the bottom of the screen, just on a little tab at the bottom. Now, the first one is the chat function. That's just a way for you to share a comment, maybe share the thanks at the end after Jane's presentation. Yeah, it's really just sort of any other comments throughout the presentation. The second one is the Q&A. So that's more specifically, if there's really specific questions that you want to ask Jane, we'll bank those up and then we'll get to those at the end of the presentation. So feel free to add questions there at any time and we'll get to those at the end. Now to try the chat function, and also for Jane and I just to get a bit of a sense about who is out there and where you're all zooming in from, I'd love you all to please open up the chat function and make sure your messages are going to be sent to everyone, and just write in where you're zooming in from. We've done this all week and it's fascinating, and so please let us know, whether it be your suburb or your state or your country. It'd be lovely to see where you're from. So many people, I love this. Wollongong, Mount Waverley, Canberra, Brisbane, Melbourne, Princess Hill, Wurundjeri Country, wonderful. Woodend, Tarneet, very good. Preston, up north, Currumbin, north, south, east, west, up from India as well. We've always had a visitor from India, so welcome as well. And Canberra, quite a few Canberrans today, fantastic. Kangaroo Valley, New South Wales, Fairfield as well, wonderful. Lots of people in today. Many, many people wanting to hear about your work, Jane, so this is great. Well, welcome everybody. Thank you so much for coming, and thank you so much for sharing where you're zooming in from. So that's it. Enjoy the presentation, everyone. I'm going to turn my screen off, and I'll hand straight over to you, Jane. Thank you so much.
Great, thank you. I'll just share my screen. And nice. So this should be working now, I hope. All right, and so in addition as well to the Q&A and chat functions after this, I've also included my email address and Twitter account if you want to ask me any follow-up questions you think of afterwards. So today I'm going to be talking about the weirdest mammals in Australia and my favourite animal in Australia, which is of course the short-beaked echidna. But monotremes also include the platypus, so I will be talking a bit about those guys as well. So what do I mean when I talk about monotremes? If my slides would change for me. All right, so when I'm talking about a monotreme, particularly in Australia, we're talking about the platypus, the short-beaked echidna, and they're in two separate scientific families. What a lot of people don't know is there's also a long-beaked echidna, which are only found in Papua New Guinea and New Guinea, part of Indonesia, and I'll talk about those a little bit as well. And of course, a monotreme is famous because they are the only egg-laying mammals. The echidna egg, for example, is very small. Platypus is similar size, and quite nicely, it's about the same size as a 5-cent coin, which just happens to have the echidna on it. But of course, they are also mammals, so as well as laying eggs, they're young, which are very tiny when first born. You can see this little one, this little jelly bean here is the new baby echidna. They do suckle milk, but they don't have any teats. The milk actually sort of gets secreted from modified sweat glands in the pouch, which is pretty crazy. And of course, being a mammal, they also have hair and are warm-blooded. So monotremes also have a couple of other characteristics which make them pretty unique. The word monotreme actually means one whole. And this is because, as this nice echidna here is demonstrating, they only have one hole, which is where all the urine, their poo, and their eggs come from, which is very unlike pretty much every other mammal. They also have a couple of other weird characteristics. So all adult monotremes have no teeth. They've lost all their teeth. They do have an egg tooth when they're a baby, and then the platypus has some teeth a little bit when it's a juvenile, but then it loses them, and the adults have absolutely no teeth. The only thing they would have is just these horny pads in their mouth, and the echidna has something similar to help grind down their food. They also have the special skill where they can detect electric fields. So both the echidna and the platypus have what's called electroreceptors in their beaks and bills, and this is how they can detect their prey, and I'll talk a bit more about that later as well. And of course, males have a spur. So in the platypus, this is venomous. The echidna, not so much, but both males do have this very impressive looking spur. And as you might have seen recently, it's just got announced last year, we recently discovered that they're biofluorescent. So if you shine a UV light on these guys, they light up like a Christmas tree. It's very just insane. And don't ask me why, because I have absolutely no idea, but it's still pretty cool. And as we'll go into in this talk as well, they also have this really unusual mix of mammalian and reptile features, which makes them a really interesting species to study in addition to their reproduction. I did want to point out though that echidnas are no relation at all to hedgehogs or porcupines. So they do look very similar. Both hedgehogs and porcupines do have these very distinctive spines, and they are made of similar material to the echidnas. But as you can see here, they look very different morphologically. They do have in common that these guys, hedgehogs and porcupines, also eat sort of ants, termites, and worms and things like that, but they are not related. This is an example of something we call convergent evolution, where two completely separate species have evolved to do a similar sort of job and function, but they're actually not related. So when we're talking about echidnas, we're talking about the group of mammals known as monotremes, and if you're talking about a hedgehog or a porcupine, we're talking about eutherians. So eutherians include us. This is just a tree of evolution from where all the current species are listed at the top, or the summary of the species that exist today are listed, and then at the bottom is the evolution from going backwards in time up until the very basic ancestors. So when we're talking about mammals, we're talking about all these three groups. So we're sorry, that was my front doorbell. Ignore that, sorry about that. So I'll wait until that stops. Okay, sorry about that. So we have the monotremes, which I'm going to be talking about today, and then we also have these other mammals which are collectively known as therian mammals, and that includes the marsupials like the kangaroo and the koala, and then eutherians is pretty much every other mammal. So this includes the hedgehog and the porcupines, but also mice and humans like us. And you can see from this picture that monotremes separated off in terms of evolutionary terms a long time before humans did. So about 184 million years ago, the monotremes separated from every other mammal. And this is just about the start of the Jurassic period. And then the marsupials and the eutherians split around 160 million years ago. So they're very distinct in terms of their evolution. They haven't had a common ancestor since then. And we know this because we have found the ancestors of monotremes. So they don't look much like platypus or an echidna, and this guy's pretty tiny. He's only about the size of a mouse probably, but he has the same sort of characteristics that we see in the modern day monotremes. And yes, this guy was around at the time of the dinosaurs. So this is interpretive artist's interpretation of a dinosaur called Bacuala alba, which was around at exactly the same time as these guys here. So in terms of the monotremes that exist and are alive today, as I mentioned, we've got the platypus, and this one is considered near threatened. It's almost, it's getting close to being endangered. But the short-beaked echidna, which is at the moment not endangered, as far as we know. And these are found in Australia, as well as the echidna a little bit in Papua New Guinea. And then, as I mentioned, there's 3 species of long-beaked echidnas, and these guys are really funky. So there's 3 species we know of. There's the eastern long-beaked echidna, the western long-beaked echidna. Oh my God, I can't believe this is happening. I have my doorbell ring today. Okay. The western long-beaked, so the eastern long-beaked echidna is probably the most common. It is found in, so I'm talking about this area. They're not found in Australia, but they are found in this New Guinea part of Indonesia and Papua New Guinea. The western long-beaked echidna is critically endangered. It's found for a very small home range. And then there's the last one is the Sir David's long-beaked echidna, and we think that one is possibly even extinct. It's only found in one very small range. It's only found at the top of one mountain as far as we know, and no one has seen a live one in recent times. These guys, as you can tell, are named for their very long-beaked relative to the short-beaked echidna, and these guys are actually nocturnal and eat worms rather than ants, so they're pretty special with that. And these guys can get to up to about 15 kilos, so they're a pretty big echidna. I mentioned we don't have long-beaked echidnas in Australia at the moment, but we might have previously. We have found cave paintings of long-beaked echidnas up in the Kimberley region, which is up in the top Northern Territory. And there also have been some specimens collected that have been listed in museums that have been collected from that area from back in the 1800s. So we might have had them in the past, but we can't find them anymore. So today I'm just going to be concentrating on the Australian species. So that's the platypus and the short-beaked echidna. And the platypus, when it was first found, when it was sent over to the England with scientists, they completely thought it was a hoax. There's a paper where they describe in detail, they actually sat there looking at the skin of the platypus trying to find stitches because they thought their friends had sent them as a joke. And this is because they just have so many weird combinations to what you can imagine the scientists would have known back in those times. So to start off with, of course, they have the very distinct duck bill, and they look like they've got these webbed feet, quite similar to the ducks. They also have what looks like an otter body, but on top of that, they then had what looks like a beaver tail. And of course, they also had these venomous spurs. What we know now is, of course, the platypus is real and does exist. And all of these features are pretty unique to the platypus. So their snake fang or venomous spur is located on their back feet, and this does contain venom, and it's very distinct. It's the venom in there is not similar to a snake, it's all its own thing. And it's also, I highly recommend don't pick up a platypus because this spur is supposed to be the most painful spur you can get. They also have their tail here is very important in terms of their health, so they use it to store fat. And this is a really good indication of the health of a platypus, is how fat and robust is its tail. Their otter body is because they have this really thick insulating fur, which keeps them warm underwater and also in colder temperatures. And of course, they have this unique duck bill, which is not really like a duck bill, it's much more leathery. And they also have in their duck bill these electroreceptors, which allow them to pick up these electrical signals to find their food. And of course, they do have these webbed feet, but I should point out that this webbing you can see in their feet is actually only while they're swimming. So while they're swimming, they just use their front feet to swim, and they use their back feet just to steer, like rudders. And then when they're walking on land, they actually pull back this webbing, so they have claws, and then they can walk, and this also helps them to dig as well. So where would you find a platypus if you wanted to go look for one? Well, for starters, they're only really found on the eastern coast of Australia and in Tasmania, and they are found around waterways. So they need a nice, clean, running water with these really, with good banks on the side of it, because of course they also build burrows. And these burrows are really, is where they sleep and raise their young. And they also can keep them at a really consistent temperature. So it's got, you probably can't read the temperatures here, but even in winter where the temperatures can range from -12 degrees to 34 degrees in summer, and the water can range from 5 to 24, the burrow maintains a temperature between 14 to 20 degrees, which keeps a really stable environment for them. And of course, they do need to live near a water source as well, because that's where they catch all their food. So they live off all these aquatic invertebrates that are found in the water, particularly yabbies and crustaceans and things like that, which you can see this one eating here. If you do see a platypus, you're not likely to see it underwater unless you're very good, but you can normally see them when they're sitting on top of the water. So what happens when the platypus feeds is it actually closes its eyes, it shuts off its nostrils, and it doesn't breathe, so it holds its breath. So the only way it can find its food is by feeling around, using its beak to find the electrical fields of the little animals moving around, and that's how it catches its food. But then to eat it, because it's holding its breath, it has to come up to the surface. So you'll quite often see a platypus swimming around on top of the surface, and what it's doing is actually eating all its food, and then it'll duck down underneath the water to feed again. Platypuses, of course, lay eggs. The platypus female doesn't have a pouch. Instead, what she does is she sits in her burrow and curls her tail around the eggs to keep them warm while she's incubating the eggs. And then once the platypus young hatch, they also stay in the burrow, where the mum will go out and get food and come back and feed them. So they're pretty cute. And they'll stay in there until around about late summer, when they'll emerge from the burrow. So these guys aren't quite ready to emerge, but they're getting close. And about late summer, and then they're completely independent, and they'll go on to live themselves. And most platypuses also will always have twins as well. So I mentioned that platypuses just recently got reclassified as being threatened, so they are in danger of becoming endangered. And this is mainly to do with issues with their habitat. So they do need a good supply of clean water and good banks, as I mentioned earlier. And so anything that affects this in terms of drought, clearing vegetation, any modification of the waterways, like this would just be not good platypus habitat. There's hardly much water left, all the banks are destroyed, so they couldn't build their burrows. And then anything that modifies the waterway, like a dam or a weir like this, can really affect their ability to live and happily. And of course, litter, and if they can't, if it's poor water quality, then their food sources can't survive either. They also suffer from predation, so foxes will eat platypuses, and they have been found to eat quite a lot of platypuses if they're around, particularly when they're moving between water sources. And there's also issues with various nets, most of which have been banned from use now, but it can still be a problem. And particularly in Tasmania, they have this really bad disease known as mucormycosis, which is also affecting platypus populations. So what about echidnas? So these are the guys that I work on the most. So they're the ones that I sort of focus on. And even though they don't look like it, they do actually resemble, have quite a lot of things in common with platypuses. So they actually do like water. They obviously don't spend all their time in it, but they are quite good swimmers and will quite happily go for a swim. They don't have the platypus bill, but they do have this long beak with the no teeth, as I mentioned, and they also have those electroreceptors, so that's how they use to find termites and ants underneath the ground. And they have these really strong forelimbs, which really helps them to dig as well. Of course, echidnas do have this long sticky tongue because echidnas, their main food is ants and termites. So they use this long sticky tongue to pick up their food and then they grind it in their mouths like the platypus does with their food. I didn't mention, but platypuses are mostly nocturnal, and so they only come out at night for the most part, whereas echidnas, you'll see them during the day. So much easier to see an echidna in the wild if you're walking around. Couple of other things with the echidnas, of course, they have these spines. These spines are actually made of keratin, so sort of similar to your fingernails or to like a rhino horn, and they're sort of modified hairs. And they actually do lose these spines. So they don't shoot them out, but they do shed them. And you can see here, there's some little spines regrowing. So they're constantly regrowing these and losing them as they go. And they do have a fur layer, quite intensive fur layer underneath the spines. They also do have ears. They don't have an ear as such, but they do have a hole where they have ears. A lot of people think they don't have ears and these little eyes. They also have a tail. A lot of people don't think they have a tail, but they do have a very distinct, cute little round tail here. You can see at the back of this little echidna with a very distinct pattern of spines. And then, as I mentioned, all the males do have a spur, but this spur is not venomous. It doesn't have any poison, and you don't tend to be spurred by an echidna. If you do pick one up, you're much more likely to get spiked by them instead. And this is a lot smaller compared to the platypus, so it's on their back foot as well. And we think they do sort of secrete things out of it, but we think it's mostly just to help them with mating and recognizing each other. So where do short-beaked echidnas live? Well, the answer is basically everywhere. So echidnas are pretty much the most widespread mammal in Australia. They're found in every single environment, right from the deserts all up to the mountains to the snow. So it's not uncommon to see an echidna in the snow. They're quite happily living in any environment and any temperature. There also is a few short-beaked echidnas living in Papua New Guinea and New Guinea as well. Most echidnas across Australia will look the same, but there is slight variations just in terms of the colour of their fur. And also the ones in Tasmania, like this guy down the bottom here, have a lot more fur, obviously, to keep them warm. And so they tend to look like they don't have many spikes, but it's more that their fur is a lot thicker to keep them warm. Something else about echidnas you might not have realised is that their back feet face backwards. So this is very unusual. Their front feet still are fine, but their back feet are twisted. So they see it here, the claws are picked up backwards. And a fun thing you can do is next time you're at a museum, go and have a look at the stuffed echidnas that they have. Because a lot of these, when they got sent over to the museum, so this is one from the Natural History Museum in London, they just got sent the skin and they just assumed that the feet faced forwards, but in fact, you can tell from these two pictures, this is actually not the right way an echidna foot should appear. and many museum specimens will have their feet facing the wrong direction. Echidnas also have very distinct shoulders, and this gives them a very distinctive waddle pattern. And this is a reptilian factor. So this is one of the things that makes them distinct from other mammals. They do have these very strong shoulder blades and rotating. And you can see the bone of the tail here as well. And when I talk about their waddle, I've got a video here to show you exactly what I mean. This organisation of their shoulders gives them a very distinctive walk. So hopefully you'll be able to see this video here. So this is of one of our captive echidnas up on the Gold Coast, and you can see hopefully this very distinctive waddle. You can also see here his little tail at the back here as well, and hopefully you can see too, this one knows that his food comes out from this door that he's walking up to here, and he can this also demonstrates that echidnas are actually very good escape artists, so they're very good at climbing as well. On top of that, their back feet as well, you might have noticed, have very long claws. So these two claws here are used for grooming. So one of their skills is they can actually use their back feet to scratch their heads. They're very sort of contortionists as well. But these back feet also have another skill in that it helps them escape from predators as well. So echidnas are actually incredibly strong. You don't sort of realize it, but this is an MRI scan of an echidna, and you can see just here how many muscles it's got on its forelimbs and also on its hind limbs as well. And this is true of both males and females. They basically look like little Arnold Schwarzeneggers almost. And this gives them the ability to very easily hide and get away from predators. So quite often if you're walking in the bush, you might see an echidna, but not for very long. They're very good at burying themselves in the ground, and they can basically just dig themselves a hole straight down in the ground. And they can do this because of these backward facing feet. So if you do see an echidna, you're not going to see one for very long because they can immediately do this. And then you can imagine if this is on a bush floor with a lot of leaves and sticks lying around, you'd have very trouble seeing this echidna. The other thing they can do is roll into a ball. So all echidnas are completely covered with a layer of muscle which all their spines are embedded to, and they're very good at folding up, and they can completely cover themselves up so that if you find them, if you try and disturb them, they'll fold themselves up tall. So all their soft bits are all covered and they just look like a little spiky ball. And believe me, these spikes are very, very spiky. They're incredibly sharp. So one thing, if you do find an echidna on the side of a road and you want to take it to a rescue, do not put it in a cardboard box. These guys are incredible escape artists. They can get through pretty much anything, and they're very strong, very sharp claws. So when we carry them around in plastic rubbish bins because that's about the only thing they can't climb out of. You might have seen recently as well, when the bushfires were happening in Australia, this photo was circulated a bit. Something else echidnas can do to get away from fire, normally they can go in their burrows, but they can also dig themselves down in the ground here. And what happened with this one is that its spines got melted. But they can actually survive fine with this because, as I mentioned, they do regrow these spines, and they're just made out of sort of like keratin, sort of like your fingernails. So just imagine just having a bit of a trim in your fingernails, and then these will grow back. So that's one useful way they can look after themselves as well. I mentioned you can also, echidnas are quite happy in being in the snow, and this is partly because they're really good at controlling their body temperature. So an echidna's body temperature is about 32 degrees normally, but they can quite happily drop this down to about 20 degrees or even lower. Echidnas that do live up in the snowy regions and also in the colder regions in Tasmania can go into a type of hibernation called torpor, which is when they drop down their body temperature very low to survive these cold temperatures for a period of time. But despite this, when they're incubating an egg, they can actually maintain their pouch temperature even when they're hibernating in torpor at a very consistent 30 degrees to keep the egg warm while it's incubating. So in terms of the reproduction, which is what I'm focused on in terms of echidnas, the reason why I'm looking into the reproduction and development of the echidna is because very little is actually known about them. So the very first paper was published back in 1887 on echidnas and by William Caldwell. But since then, only very little has been done. So there was one paper that looked back in 1894 about the development of the young. But that's pretty much been the only thing that's been done. Since then, there's one paper, a couple of papers in the 1940s looked into their ovaries and development of the eggs. And Mervyn Griffiths is sort of the father of echidnas, and he did a lot of research into echidnas, but didn't have many samples in terms of their reproduction to look at. So we really haven't learnt much more than these earlier studies back in the late 1800s. Part of the reason for this is just the challenges in working with echidnas. So as I mentioned, they're very good at hiding, so they're very difficult to actually follow them around and track them. And quite often you basically won't see, most of the time you'll only see an echidna by itself. It's very rare to see a group of echidnas. The only time this does happen is when it's the mating season, when one female can get followed around by a whole group of males trying to mate with her. And these are known as those echidna trains you might have seen. Where one female has been reported to be followed around by about 11 males. But most of the time, these guys are solitary, so they're always spending their time by themselves. Another challenge with working on echidna reproduction is it's really difficult to tell the difference between a male and female echidna. So there's basically no size difference between males and females. They're both about the same size. If anything, females are a little bit bigger. The females do have a pouch, but this only develops when they're incubating. So these pictures are both of a female, one outside of the breeding season and one in the breeding season where she's developed her pouch. And this is really a pity because they do have such amazing reproduction in terms for mammals. So they are the only egg-laying mammals. And then on top of that, they do have some other really weird reproduction things. So one of these is their sex chromosomes. So in humans, if you have an X and a Y chromosome, you're a male. And if you have two X chromosomes, you're a female. And this is because males have one particular gene on their Y chromosome that makes them male rather than female. Echidnas, however, have 5 copies of these X chromosomes and 4 copies of the Y chromosomes, and that's what you need to make a male. And females have 5 and 5 X's, so 10 X chromosomes in total. But they're missing the gene that makes them male, and we actually don't know why they become male rather than female at this stage, and that's one of the things I'm looking into. But I don't have an answer for you yet, unfortunately. And the other major reason in terms of looking at their reproduction development is just because we had real trouble trying to breed them in captivity. That is until now. So I work with Currumbin Wildlife Sanctuary up on the Gold Coast, and they were pretty much the first zoo to crack the code of how to successfully breed echidnas in captivity. So they've now had a successful breeding program going for at least the last 5 years and have a colony of about 30 echidnas up there that we work with, and this is one of their babies that they had up there. And echidna, as the colony we work with, there's about 30 echidnas, and they have specialised nesting boxes and things to work with. And they're also under 24-hour video surveillance, so we can get some really good information about when they're laying their eggs and things like that. Currumbin also has a wildlife surgery there as well, so we're able to do things like anaesthetise the echidnas to check on their eggs and their babies, which you can do just by putting a tube over their snout. It's also really easy to take a blood sample from an echidna. You just need to, they've got a blood reservoir at the end of their nose, so you don't need to find a vein, you just need to put a needle in their nose when they're anaesthetised, like this one is. And you can also do things like ultrasound, which is also needed to tell the differences between males and females, at least in the adults. So part of the reason that Currumbin had such breeding success was due to a couple of reasons. One, obviously, was finally actually putting males and females together. A lot of the early zoos just didn't have a way to tell them apart, and so we're probably putting 2 males and 2 females together. We've also discovered they need a really high-fat diet, so we just basically weren't feeding them enough. And this is partly because they do eat ants and termites. You can't replicate that in the zoo environment. It's like you can't provide enough ants and termites. So we have to supplement their diet with a mixture of mincemeat, but we have to add extra olive oil to that to make sure they have a high fat enough diet. And then they get a termite nest as a treat once a week. We also discovered that even though they're up on the Gold Coast, they still really like to be warm. So we do have heat lamps for them. And as I mentioned, they are under 24-hour video surveillance. So that's been really useful for our study as well, to know exactly when these guys are pregnant so we can make sure to give them extra food. In terms of a lot of the samples that I've been working with is because Currumbin, as I mentioned, does have a wildlife hospital, and unfortunately about 50 echidnas per year are brought in injured in road accidents into Currumbin Wildlife Hospital. And this is especially in the breeding season, which is about now, so July to October, because this is when the echidnas are on the move trying to find a mate. If the hospital staff can fix the echidnas, they will. But unfortunately, what happens a lot of time when an echidna gets hit by a car is that it breaks its beak. So this is a CT scan of an echidna showing the bones, and you can see here with its skull, that beak you see actually has some very fine bones in it. And you can imagine if an echidna gets hit by a car, it's a high possibility that it's going to break this beak. And unfortunately, because they rely on it so much for their eating, you can't really put it in a cast, and there's just nothing we can really do, and they have to be euthanized. But that has allowed us to collect some tissues from these guys, which we've been using in our studies. So one of the first things we've been working out is actually how long is pregnancy in the echidna. We really had no idea until this time, because it's quite difficult to follow an echidna or even know if it's pregnant. So with the combination of using hormones and ultrasound, we've now been able to work out that pregnancy in the echidna lasts about 18 days. So this is the time when the embryo is in the uterus. At the end of these 18 days, they lay their egg, which I mentioned is only about 1.5 centimetres in diameter. And then they incubate their egg in the pouch for about 10 days until the egg hatches. Something else which has made echidnas difficult to study too is during this pouch incubation, what the mother does is actually stays in her burrow incubating the eggs, and she doesn't leave at all to feed or to poo or do anything during this time. So this is also, if they're in the wild, it's very hard to know how long this period lasts for, because they just never emerged. Once they've hatched, they then go into the pouch in the echidna, and they stay as a pouch young for around about 45 to 55 days, because after this time they start getting too big for the pouch, and they start growing spines. So as you can imagine, the mum boots them out, and from this time point onwards they're left in the burrow. So they then become staying in the burrow, where they're known more as a puggle, where they grow up and stay around about another 150 days until they become a fully formed little mini echidna, and they're perfectly independent when they're weaned from this point onwards. So in terms of echidna eggs too, you might have noticed from my previous pictures, if you were watching very carefully, is that the echidna egg doesn't look like what you might expect. So it's actually quite a leathery egg. A lot of people think that it's going to be more like a chicken egg, but you actually can't crack an echidna egg as such. If anything, you're going to squash it. So these are much more leathery, they're much more gentle, and you can tear them. This is a brand new echidna that's just hatched, so this is why he's got a little bit of blood going here, and you can almost see a little egg tooth there as well. And these eggs are actually much more similar to what you see in some lizards and snakes, where it's a much more leathery egg and much softer. Something else with these echidna eggs too is that we've been having a look at their ovaries, and in terms of when you're looking at ovary development, if you're looking at like a normal mammalian ovary, so this is a cow ovary, but human ovaries look pretty similar. This is sort of what the ovary looks like, and the eggs are in these little clear spaces here, whereas the chicken egg looks much more distinctive. It looks like they've got these big yolky eggs here, and it's much bigger compared to the size of the cow ovary. And when we have a look at the echidna ovary, they look much more similar to the chicken ovary. And this is because all of these animals that lay eggs need yolk. So this is what makes the size of the eggs a lot bigger in the echidna, or the size where the eggs are located in the echidna ovary so much bigger, because this is all the yolk that the young needs to feed off when they're in the egg. When you do with animals that do have yolk, you also need a protein called vitellogenin, and this is one of the proteins that provides a lot of the nutrients and things when they're in the egg. And we knew previously from other people's studies that chicken have 3 types of this protein. So they have 3 types of vitellogenin, which means they can create quite complex yolk and nutrients. So we wanted to look at what about monotremes, whether they also have this vitellogenin protein. So to do this, we recently had access to the echidna and platypus genome that got published. And from this, we can do a study like this, where what this is showing is one section of a chromosome, and all the different coloured boxes are different genes. So if it's a different colour, it's a different gene. And so the top lists the human, all the genes listed in the human in this particular region of the chromosome. And then we can find these genes in the other regions of other species like the koala, platypus, echidna, and chicken to see which genes they have in common and which ones they don't. So animals are actually a lot more similar than you would imagine in terms of their genes. And if they have a similar, so for example, if the koala has similar genes around these same locations, but they're missing one or two, then that means that it's pretty likely they have lost these genes. So the first thing you can notice here is that chicken in this particular region has pretty much all of these genes, including the 3 copies of the vitiligenen here. So the VTG, I've labeled them here. So it's VTG 2, 3, and 1. What you can see if you go up from here is that humans have lost all of them. They don't have any copies of these vitiligenen genes. But you can see that platypus and echidna, they definitely have a copy of the vitiligenen 2. They've lost the copy of vitiligenen 3. And they have, we can tell that they have a version of vitiligenen 1, but it doesn't appear to be functional, which is why I've put a dotted line around here. So they appear to only have 1 copy that works, and they've lost 1, and 1 appears slightly. They still have it, but it probably doesn't work anymore. What's interesting about this is you can also see that the koala, while it doesn't have 2 copies of the gene that the platypus and echidna might have, it does have a copy of the vitiligenen 2, or at least a copy that we think must have been functional in the past, because they've got enough of it to recognize it, but not enough for it to actually work. And this is really interesting from an evolutionary point of view. Okay, the areas that I've circled. So 2 questions. One is, why does the koala still have a version of this vitiligenen when they definitely don't lay eggs anymore? And why doesn't the echidna need all 3 copies of vitiligenen like the chicken? Because they do lay eggs, but they've only got 1 copy left. And this is to do more with how the echidna, how the echidna's reproduction works. So in the chicken, they rely entirely on the eggs for their nutrients, whereas the echidna, even during pregnancy, when the egg is in the uterus, they actually have the uterus helping them and giving them secretions and nutrients, as well as the yolk in the egg. And so they only really need their yolk in the egg for this 10-day, short 10-day period when they're actually being incubated in the pouch. And then, of course, after that, most of their development is happening when they're in the pouch and in the burrow, when they're feeding off milk. So echidna milk is actually really high in fat, and that's partly why we think they need a high-fat diet to make sure their breeding success. And they drink a lot of milk. So an echidna baby of this size, when it's first born, is pretty tiny, as you can see here, drinks a lot of milk. So this white ball here is all the milk, in its stomach, and they can drink about 10% of their body weight in milk at each time. They also grow pretty quickly. So to get from this size to this size only takes about 20 to 30 days. So that's a lot of eating to do to get to that size. And then the other thing that happens is that once they're left in the burrow, the mum doesn't feed them every day. So once they get to about this stage, they're only getting food about once a week. And this is really different compared to something like another marsupial species like the tama wallaby, where even though they start off the same, so this is a newborn tama wallaby, or close to, when it's in the pouch, and you can see it's pretty similar in size to a baby echidna. And like the echidna, they also feed off milk and have a really long period in the pouch and time when they feed milk. But unlike the echidna, the wallaby is never left in a burrow. It's always in the pouch and always has a constant supply of milk. So their milk doesn't have to be so high in fat. Whereas the echidnas really do, because they just very few times, they don't get milk anywhere near as often, and they have to survive a lot longer. So they, and we think also that termites and ants are really high in fat too, which is also partly of this reason. So their milk is just a whole lot fatter, basically, than what you need to keep a wallaby PY alive. So what does this mean in terms of evolution? I mentioned it's really interesting that even though marsupials don't have an egg, they do have this copy of the egg protein. So we can tell, this is just the evolution diagram again, and we can see here that all these species on this side all have eggs. So that's established. We also know that both monotremes and marsupials have a really short pregnancy, but then most eutherians have a much longer pregnancy. So marsupials also only have about 20 days to 25 days where they're actually in the uterus, versus humans up to about 9 months. We know that echidnas also rely heavily on milk. As I mentioned, marsupials also rely on milk, but only and have a copy of this egg protein, but they don't actually have eggs anymore. And then we know that humans don't have any of these egg proteins, but they do have milk. If you look at the evolutionary tree, this means that even though humans don't have a copy of this egg protein, because both marsupials and echidnas both have a copy of the eggs and they both have milk, this suggests that our common ancestor back here did have a copy of eggs. So this does suggest that originally our common ancestors did lay eggs, and in the meantime, we've moved on to having a longer pregnancy and using milk instead. So quite likely that these, and because the marsupials also still have a copy of the egg protein, it's quite likely even these ancestors. So maybe your ancestor 160 million years ago might have laid eggs. And then finally, I just wanted to put this up too. I've got colleagues over at the University of Adelaide who have a citizen science app called Echidna CSI, and this is to help us find echidnas. So I mentioned echidnas are found all around Australia, but we actually don't know how many of them there are. So this app was developed so you can record your echidna sightings, and they've got up to this was a couple of months old now, but they've had over 10,000 echidna sightings. So you can submit both, anytime you see an echidna, you can submit your photo of an echidna and record it up to this app. And this really helps us knowing where most of our echidnas are located. You can also upload a scat submission or one of their poos. So echidna poo is pretty distinctive. It looks sort of just cylindrical, but what you will see is it's sort of been described as sparkly poo. Because echidnas eat so much sand most of the time, along with all their food, because that's where ants and termites tend to be, they tend to have a lot of sand in their poo as well. And so this shows up as being sparkly. But if you have a look at the app, they've also got a Facebook page. You can get a lot more information about how you can help us out with that and try and find out, make sure echidnas aren't endangered and that their populations stay healthy. So that's all from me. Hope you enjoyed the talk. If you have any questions after this talk that you think of, you can contact me on my email address, or I'm also on Twitter. And I'd just really like to thank everyone here that I've listed at the University of Melbourne, the University of Queensland and Currumbin Wildlife Sanctuary who have been involved in this Echidna project to date, and also our funding bodies from the Australian Research Council. Thanks.
Thank you so much, Jane. That was so fascinating. There was just so many things that I thought I knew things about monotremes, but I clearly did not. And they are incredible animals. And I think it was Cooper that said that their new favourite animal. And frankly, Cooper, I think I would tend to agree with you. I think they're amazing. Now we are almost just past 2:45. So I just want to let everyone know that we will be going over today. So if you have to leave now, thank you so much for coming. And there will be recordings of this sent out to anyone that's registered. But there are so many amazing questions that we'd love to get to. So we will keep going for another 10 or so minutes just to answer all those questions. Jane, if you're happy to stay with us up until about 3 o'clock.
Yeah, perfectly happy. Okay. Sorry, that's my fault for going too long. Feel free to email me if you have any questions and you have to leave now.
There's so many great things. So, okay, so right, let's get to it. So where are we going to start? I think there's quite a few questions about sort of where they can be found. And I know you did put the map of all of Australia, but I think there was a few comments from Caitlin Hughes actually of seeing them in Melbourne and whether they're quite common in Melbourne. Are they? Would you find them quite often around Melbourne?
I'm not entirely sure. I have had people send me, I had someone just today send me a photo of one that they saw in Melbourne. So they're definitely around, but they are pretty cryptic. They do tend to hide around, but if you're around sort of the bush areas, they're definitely like up in the Dandenongs and places like that. So yeah, they do need a good supply of ants and termites and do sort of need a bush location. Yeah. But yeah, if you're around any of those areas, you can definitely see them in Melbourne.
Yeah. Oh, very good. And so how much do they usually weigh? There was a couple of questions about that from Caitlin Shepherd, actually.
Yeah, so your average echidna is about 5 kilos.
Okay, got it. 5 kilos. Trying to think of what that would equate to. They're reasonably heavy then.
They're reasonably heavy, but yeah, both males and females weigh about the same. So yeah, this small one would be 4 to 6 kilos is sort of your average echidna.
Okay. Okay. Brooke, another really quick one. So echidnas fast? I guess when we saw the video of them waddling, so they wouldn't really get up to very fast speeds, would they?
Not particularly, no.
Yeah.
I've never really seen an echidna sprint anywhere.
No, I don't think so. Thank you for your question, Brooke. And I know you mentioned as well, I think that you'd seen them around. So very, very lucky to be able to see them as well. Now, a couple of people also asked about, is it platypi or platypuses? And I think you said platypuses. So just to confirm, the plural of echidna, plural of platypus.
Yeah, I've had many arguments about this. It's platypuses.
Okay. Yep. Very good. Very good. Okay, back to echidnas. So I guess their temperament. So are they friendly? Is it sort of easy to sort of catch them when you're up doing the research with them?
So mostly I work with the captive ones. The keepers are quite good at picking them up, and quite a lot of ours are pretty tame because they've been sort of conditioned to be handled quite a lot. But in general, I mean, they're not going to try and bite you or anything. They actually can't. If anything, they'll just roll into a ball. The one I was holding most of them I can't hold. Most of them will just wriggle around, and those spikes are really painful. But the one I was holding is one that's particularly tame and used to being handled, so he's pretty happy being held. But most echidnas, yeah, they're not too happy about it.
Okay, great. While we're talking as well, I might just get you to stop sharing your screen, and we'll bring that up just towards the end when we're reintroducing. Awesome. Okay, so many other questions. This is so great, everyone. Okay, over to Vicky. So I think you talked about the spines and how they can regrow, and that amazing photo of the spines being melted, I think you said.
Yeah.
So how long do they grow? Like, do they continue to grow, or they just
they just
stop
continue to
after
grow?
Yeah.
They're basically modified hairs, so they kind of like keep growing like your hairs, so they'll fall out and regrow. Most of them will stay, but like, yeah, you can definitely see parts of them when they fall out and things. Yeah.
Mm-hmm. Now there's a couple of people talking about their backward feet, which frankly was one of my most fascinating parts. I can't believe their feet are backwards,
really
or not backwards for them, I guess. Maybe we have backwards feet to them.
Yeah, they're backwards feet. Yeah.
So I guess sort of a little bit more about why. I know you spoke a little bit about, I guess, the burrowing. Is that the main reason for the backwards feet? What would they be used for?
Yeah, we're not entirely sure why they have backwards feet. I should mention platypuses also have backwards feet too. It's hard to find a picture of a platypus. It seems to be a monotreme thing.
Yeah. Okay.
But at least with the echidnas, we're not quite sure why, but we do know having those backward feet and those really long claws really does help them do that burrowing down where they can just basically dig straight down.
Yeah.
That's pretty hard to do without that arrangement. So yeah, that seems to be why they've got it.
Yeah. Oh, it's fascinating. I love to see a video of them just burrowing
down. I know, I need to get one. Yeah.
And so can they walk backwards with the backwards feet?
No, it's just their feet are backwards. So they're walking forwards with backwards feet.
Sure.
They're twisted. Yeah.
Well, there you go, Danica. That one's for you. I think there was a few questions about the what do they call the spurs? I think on both the animals, and I guess and sort of how dangerous are they if we do get spiked by the spurs? What would be what would happen?
Yeah. So I mean, echidnas are fine. They don't actually have any. They've lost the venom. They don't have any venom anymore. So they're okay, and they're not likely to spike you. It's more just sort of a remnant thing they've got left over. Platypuses, so it won't kill you, but apparently it's very, very painful. So it's not poisonous to us as such. Like it's not like a snake bite, and it's only on the males. But if you do get spiked, it's more that I've just heard it's a really intense pain, and the area will just go numb for a bit. But,
okay. So not ideal.
You might need some hospital, just more treat the pain and the effects, but it won't. Yeah, it's not going to hurt. It's not going to kill you. Okay,
I didn't realize there was a danger factor in echidnas. Maybe this is even more interesting.
Well, yeah, that's more the platypus rather than.
Right. Okay.
Yeah.
Getting so confused. Okay, a bit of a long one. So from Caitlin Hughes. So a bit more about the echidna. So the eggs, and so they're quite small. So the question was sort of how do mothers. How long do they take to sort of have them, and are they in pain? And how do the echidnas and I guess maybe a little bit more about how they even survive when they're just that small. What was it, 1.5 centimeters?
Yeah. So the mum's pouch is it's kept really moist, and as I mentioned, it's kept really warm as well. So it is like a nice little incubation chamber for it. In terms of, we do have a video of an echidna laying an egg, but it's pretty rare. They always tend to do it at night, which is really really not considerate of them. So what they do is they kind of like just basically, they're really good at bending as well. That's how they can roll themselves in a ball. And so when they're laying an egg, they basically curl their tail over and lay the egg directly into their pouch.
Oh, okay, right, yeah.
So yeah, it gets flopped straight into the pouch. Yeah, it doesn't take that long. It's not, I don't think it's difficult to lay an egg. They're pretty small. And because they're so soft as well, it's probably not such an issue. And once they're in the pouch, they stay in the pouch, and the mum doesn't move during that time too, which would also help. So it doesn't get damaged probably as well. And they incubate them for about 10 days, and then they hatch, and it's the pouch young.
Yeah, and good reminder about the eggs. I guess whenever you talk about an egg, I just always think it's that hard shell. But
I know everyone think it's a hard shell. It's really not. Yeah,
no, that's fascinating. What else can we go? Oh, from Lockie. What are the main predators of echidnas?
Someone asked me this the other day. They actually don't really have many.
Oh, okay. Yep.
It's more. I know dogs can attack them and things, but because of their spikes, they're actually really hard to get into. One of the only reports I've heard is like a dingo was getting one, and what it did was actually drowned it. Like it was by the beach, and it took it to the water and drowned it so that it could get to it. Because they're extremely strong. Like the first thing, if you want to like to get them to put an anesthetic on them, they're really strong, and you actually literally have to like bend them apart, sort of thing, to get it. So it's pretty hard to undo them once they're in one of those balls.
Oh, okay. Yes. So, fascinating about those balls.
They can do it, but if you have foxes, dingoes, that's pretty much it.
Okay, ready to go. Well, along with the echidnas then. One from Jennifer, quite interesting. So can their ability to have twins be harnessed to increase fertility and reproduction in other animals where twins are preferable, like lambs? I don't know twins are preferable in lambs, but I mean, it's an interesting question.
But you get two for the price of one.
I guess any animal.
I actually have no idea. Echidnas can have twins as well, but it's not a frequent occurrence, whereas platypuses I know do. I have to say I don't know. I don't think anyone's tried to look to see how they do it. Yeah. It's very hard to get platypus samples to look at things like that too. Yeah. So it's probably quite difficult.
Sounds like it. I mean, it sounds like we're just at the start of really understanding the reproduction of these animals, which is just fascinating. And so great that you're able to do this work. So good one, Jennifer. Maybe there you go.
There you go. Something else to look at.
Yeah, exactly. Something to do. Okay, we've only got a couple minutes left. So a couple last questions. How long do they live for?
A long time.
Long time.
Not sure about platypuses, but there's reports of echidnas living up to 50 years.
Rightio. Okay. That's amazing.
There's echidnas in Currumbin we think are at least 30 years old because they've had them for quite a while and they were adult when they got them. Yeah, they can live a long time.
Good on you, echidnas. Love it. Yeah. Okay, great. Well, look, we've only got a couple minutes left, so I might just throw one last question to you in terms of your research. So what's sort of coming out? There's a lot of things you've worked out about the reproduction. What's the next sort of key thing you'd love to find out?
We're really having like a whole lot of different things. So if nothing else, we're even just looking at like their reproductive tracts and things as well, because even stuff like that, we know the uterus helps the eggs grow, but we're really not sure how. So that's one of the things we're looking at in terms of just having a look at the uterus. And I know that area from my pregnancy research, and just their development in general. There's just a whole lot of projects we've got going on at the moment. Pretty much anything you can think about reproduction and development we're looking at the moment. So it keeps me busy.
Fantastic. Well, this was fascinating. And there were so many comments in the chat. Hopefully you caught a bit of it. And there was a lot of grateful audience members just hanging on your every word. So thank you so much. Before I sort of finish off, I just want to let everyone know out there, number one, thank you so much for joining us and for all the great chats and all the amazing questions. There will be another mid-afternoon masterclass tomorrow. It's going to be all about vaccines. So that'll be a really interesting chat. That's with Dr. Jennifer Juno from the one and only Doherty Institute, which has obviously done a lot of work over the last couple of years. This event also will be it's been recorded and it'll be on YouTube in sort of the next couple of days. And if you've registered for the event, we'll be sending that link out to you as well. I think that's it. Jane, maybe if you want to share your screen just to put the information up about the event tomorrow, if you have them. Yep, if you could, fantastic. So there you go, everyone. That's the more details about the event tomorrow. If you can join us, it'd be wonderful to have you along. I usually say see you at this point, but I won't be able to see you, but you'll be able to see us and our guests. So finally, Jane, thank you so much. It was a fascinating talk. Thank you everyone out there for joining us. I hope, that was everything you hoped for. For me, it was so much more. I learned so much. So thank you so much. And we will see you all tomorrow.